Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Using CSF Biomarkers of Alzheimer's Disease in Early Clinical Development. My name is Margot, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank QPS, who helped develop the content for this presentation. QPS is a global contract research organization providing discovery, preclinical, and clinical drug development services since 1995. Their mission is to accelerate pharmaceutical breakthroughs across the globe by delivering custom-built research services. An award-winning leader focused on bioanalytics and clinical trials, QPS is known for proven quality standards, technical expertise, a flexible approach to research, client satisfaction, and turnkey laboratories and facilities. They are passionate about their work, creating value for patients and customers while generating opportunities for employees to thrive. Now I'd like to introduce our speakers for today's event. Isaac Dendas is an experienced clinical pharmacologist with a demonstrated history of working in the pharmaceuticals and contract research organization industry. He brings strong operations professional skills in medical devices, biotechnology, cell culture, research and development, and CRO management. His current responsibilities focus on ensuring that the QPS Netherlands patient clinics are functioning seamlessly, including management and maintenance of the quality system, defining and managing the procurement policies, as well as scientific support by evaluation and design of patient studies. Isaac also supports QPS Netherlands business development in the sponsor acquisition process, identifying the wishes of the sponsor concerning patient studies, gathering and evaluating study feedback, participation in sponsor visits, and taking the desired steps for improvement of the quality of patient studies. Next, we have Professor Peter Paul Dendane. Professor Peter Paul Dendane is a neurologist and neuropsychiatrist at the University Medical Center Groningen, specializing in preclinical and clinical studies of neurodegenerative disease. Professor Dendane is the scientific director of the UMCG Alzheimer's Center Groningen. The Alzheimer's Center Groningen is a leading knowledge and research center in the field of Alzheimer's and dementia. Their objectives are to improve the quality of life of people with dementia and their close relatives and to stimulate scientific research into memory and disorders in dementia. So without further ado, I'd like to hand the presentation over to our speakers. You may begin whenever you're ready.
Well, thank you very much for your nice introduction. So I'll give a brief presentation on biomarkers of Alzheimer's disease. It will be a little bit broader, not only inclusive cerebrospinal fluid biomarkers, but biomarkers in general. Next slide, please. The content of this presentation is as follows. We will start with a small introduction, focusing on AD, Alzheimer's disease, as a particular condition, of course. We will define briefly criteria of biomarkers. We will discuss and list the type of biomarkers for dementia. Then we will actually briefly evaluate the diagnostic performance of biomarkers in AD, and their performance in terms of differential diagnostic performance of these biomarkers. We will discuss biomarkers for early, eventually pre-symptomatic or prodromal dementia. We will discuss biomarkers for monitoring disease progression, which is of course potentially important in this type of clinical trials. We will briefly also mention that biomarkers can be used for enrichment strategies in randomized controlled trials, and biomarkers can, of course, be used as surrogate outcome measures in AD trials, a phenomenon that takes increasingly place. And of course, we will draw some final conclusions at the end of my short presentation. Next slide. It's of course important to reach an etiological diagnosis of your dementia syndrome, because dementia is a general umbrella, and of course when we target specific disease pathophysiological aspects, we like to target an homogeneous patient population, and quite often in the clinical research we are performing in the field of AD, we actually want to enroll, we want to include subjects with, for example, mild cognitive impairment due to AD or early stages of AD. Next slide. We are all very much aware of the fact that neuropathological changes present in AD over a course of several decades, and many years before the first clinical symptoms are observed, there are neuropathological alterations consisting, of course, of tau accumulation, the neurofibrillary tangles, and a beta accumulation in the beta amyloid plaques. So it's quite a challenge to detect the disorder at early stages, eventually pre-symptomatic or prodromal stages. Next slide. Criteria for AD biomarkers are diverse. Here you find the criteria defined by the Reagan Research Institute, and ideally, of course, a marker should be able to detect a fundamental feature of the Alzheimer pathology. It should be easily accessible, blood, urine, or, for example, cerebrospinal fluid, or imaging techniques which are not invasive. One should look for sensitivities and specificities of 80% or above, and the biomarkers should be validated in patients with autopsy-confirmed dementia diagnosis. Later on, you will see a slide addressing that issue. The biomarkers should be useful for disease prediction, useful for monitoring disease progression, as we mentioned already earlier, and it should be have a potential in terms of monitoring treatment effects. And then we talk, among others, about surrogate outcome measures. Next slide. There are a variety of biomarkers that we use currently in clinical practice, but of course in clinical research. Some involve neuroimaging, where we look at structures, for example, MRI of the brain, hippocampal volumes, where we look at functional imaging like functional magnetic resonance spectrometry. And in addition, we can use, of course, PET imaging, which can be metabolic imaging, FDG PET, beta amyloid imaging, or tau imaging, which we will show later on. We can perform also receptor studies in these specific populations. SPECT can also be used. In addition to that, we would like to mention the CSF biomarkers, but also the advent now increasingly of plasma biomarkers that are not fully validated yet. Those CSF biomarkers have actually introduced a change in clinical approach and a change in research diagnostic criteria. One can, of course, also apply genomics, proteomics, metabolomics, electroencephalography, and magnetic encephalography can be another useful tool. Next slide. An update on the diagnostic criteria in Alzheimer's disease. Next slide. During the last 20-30 years, we have been witnessing major progress being achieved in the field of biomarker development, and currently those are applied in the clinical and research diagnostic criteria. And while in the past the neuropathological observations were the golden standard, as shown here with the amyloid plaques at the left and the neurofibrillary tangles at the right, we do not have to wait anymore until disease of the patient, and we can make an in vivo, quite accurate diagnosis. Next slide. Yeah, this is the classical figure illustrating where we were actually focusing on during the last 20 years. We look here at the NIN-CDS ADRDA criteria at the right, where diagnostic criteria were used for dementia. But we know, of course, that treatment should be started much earlier in the disease development, eventually in the preclinical state, but certainly in the prodromal early state, which precedes, of course, the dementia stage of Alzheimer's disease as a disorder. And indeed, it is possible at this moment to make a diagnosis much earlier, as we will see also in the recently developed National Institute of Aging Alzheimer Association criteria. I say recently developed, but actually they date already from 2011 and are continuously refined. Next slide, please. Yeah, here you see the MCI concept, the mild cognitive impairment concept, which has also been introduced in the DSM-5 currently, of course, and we distinguish on one hand normal aging between then mild cognitive impairment and then the dementia stage. Important to know is that mild cognitive impairment can be a reversible condition, can remain stable, or can be a prodromal condition of a dementia syndrome. The dementias that can develop from MCI are quite diverse. Not only AD, it can also be vascular dementia, it can be Parkinson's disease dementia, it can be frontotemporal dementia. The focus as of today is rather on AD. We know that there are known annual conversion rates for normal aging between 1 and 2%, for MCI 10 to 15%. The important message now is that using additional biomarkers allows us to predict conversion from MCI to dementia in one of the frequent forms, like, for example, the amnestic mild cognitive impairment. Next slide, please. Yeah, here a first paper from our group. I always make a joke and say I refer to those papers because nobody else refers to it, but that's of course not true. But these were one of the first papers actually where the performance of the CSF biomarker panels were actually validated in large autopsy confirmed dementia populations. And indeed, large groups of subjects were evaluated using neuropathological criteria and CSF biomarkers, and we were able to actually define the high sensitivity and specificity of those biomarkers. Needless to say that we talk here about combined biomarkers involving a beta 140, 142, tau, total tau, and of course also phosphorylated tau. Next slide. In addition, of course, one had to demonstrate that there was an added value that besides the neuroimaging, the neuropsychological performance and the other biomarkers, those cerebrospinal fluid biomarkers contributed to a more precise and refined diagnosis, and that we have shown, among others, in publications in Neurobiology of Aging from 2010, and some other papers clearly demonstrated this. Next slide. And this is indeed an important issue also. We refer here to a Lancet Neurology paper from Hansen, where it is shown that once you have a cerebrospinal fluid biomarker profile that is compatible with AD or predictive for AD, in subjects with a mild cognitive impairment status, you see that the odds ratio of converting to dementia increases about 17.7-fold as indicated here. So extremely high predictive, and this actually led to the diagnosis using the National Institute of Aging Alzheimer Association criteria, where MCI then received a label as MCI due to AD, for example. Next slide. Okay, here you see the different biomarkers. We start actually from the normal condition at the left, the pre-symptomatic phase. Then we have the early mild cognitive impairment phase and the light mild cognitive impairment phase, reaching then the dementia phase. You can see that at the left, CSF and amyloid imaging are very early sensitive biomarkers which are already altered before there is a change in FDG metabolism or in hippocampal volume as shown on the MRI, and before, of course, the cognitive deficits and impact of activity of daily living. You also see a line indicating cerebrospinal fluid, the tau, which accumulates steadily at a later stage of the disorder. And those specific biomarkers will be dealt with in the following slides. Next slide, please. Okay, yeah, this is a little bit summary, summarizing the Alzheimer disease pathology, the ATN classification as such, indicative of amyloid deposits, neurofibrillary tangles, and neuronal injury or loss demonstrated by the total tau accumulation. We have no image at this moment.
Yeah, that is me.
We are trying to get it again.
It's coming.
Okay, perfect. Next slide, please. Okay, so of course, early prodromal biomarkers can be used as circled here on the left, cerebrospinal fluid and amyloid imaging. Next slide, please. Okay, so actually we had a major transition from 2010 to 2011 with the introduction of the National Institute of Aging and Alzheimer Association criteria, where there was a major shift. Before we had the clinical entity of mild cognitive impairment. Clinically, we made a diagnosis of possible or probable AD, and actually it was confirmed on post-mortem neuropathological observations. That shift is represented in next slide, where we do not anymore apply a clinical pathological definition, but a clinical biological definition, and where indeed the inclusion of those biomarkers indeed allow us to indicate pre-symptomatic, prodromal, typical or atypical dementia cases related to Alzheimer disease. So these are the criteria published in Alzheimer and Dementia in 2011. Next slide. By the way, the impact factor of that journal increased dramatically due to the publication of those papers and reached a number of about 17. So I mentioned already to the NIH and NAA criteria. There were a series of consensus meetings which actually led to a series of publications which I mentioned already, 4 in total. Next slide, please. Yeah, here you see the first introductory paper by Jack and co-authors, Introduction to the Recommendations from the National Institute on Aging and Alzheimer Association, and then further papers actually focused on the dementia definition, on the MCI definition due to AD, and also on the pre-symptomatic criteria of Alzheimer disease. Next slide, please. Okay, a little summary. Main elements that were introduced in the diagnostic decision tree, let's say, were the amyloid positivity, which was actually observed by PIP or floor beta P positive imaging, decreases of A beta, and on the other hand, neuronal injury based on FDG PET and increased cerebrospinal fluid tau levels with, of course, the specific structural alterations on MRA. Next slide, please. Okay, so here we are now looking at further stages of the disease, indicators actually of disease stage, and we are focusing now on early mild cognitive impairment and later mild cognitive impairment syndromes. Next slide. So what we will be doing now is briefly give a little review of some of the biomarkers that have been used over time. What you see here is MRI, coronal slices, and you see mesotemporally the hippocampus, which progressively presents an atrophy, and these are the so-called mesiotemporal atrophy ratings, going from 0 to 3 or 4, depending on the stage of the disorder. Next slide. Yeah, here, for example, you have the volumetric analysis of certain structures, entorhinal cortex, hippocampus, and this volumetric analysis are now subject of automatic or semi-automatic analysis that can be used as outcome measures for therapeutic interventions. Next slide. Yeah, here very briefly an example of FDG glucose metabolism with normal aging at the left, at the middle typical Alzheimer profile, and right a peak or specific frontal temporal lobe pattern. Next slide. Okay, here, of course, introduction to the in vivo amyloid imaging. And this is an example of the Pittsburgh compound B imaging, where you see the column with the AD subjects, where there is actually an imaging of cortical accumulation of the amyloid in those subjects. Next slide. Well, this slide shows actually a similar observation, where actually from top to bottom you have an increased involvement of the AD pathology with an increased accumulation of beta amyloid, so disease stage dependent accumulation. Next slide. Okay, well, we should not forget tau, of course, tau accumulation, which is shown here on this slide, next to amyloid beta completely at the right, and in the middle, the tau accumulation. Tau accumulation, which is presently used in several of the trials we are conducting here in our center at the moment. Next slide. Okay, well, here another potential surrogate outcome measure is the density of synaptic vesicles, of synaptic related proteins. Here, for example, synaptic vesicle glycoprotein, and you see on the images on the second row actually there is an indication of the mesotemporal lobe with activity in the hippocampus, which is also disease dependent. And for example, this can be used in as a surrogate outcome measurement in an epigenetic treatment strategy. Next slide. Well, yeah, okay. Look, here you have another slide. Today we heard some news of related to aducanumab, which was developed in the indication of AD. You see at the left the decrease of the A-beta accumulation after 1 year of treatment, which happened actually in a dose-dependent manner. You see from placebo to 10 milligram per kilogram. You know that the development was stopped in March, but today we heard some promising news that reanalysis took place, and that potentially there are some positive alterations with regard to the efficacy of this treatment. Next slide. Okay, some additional potential surrogate outcome measures can be discussed. We discussed already several of them. We are going to give a couple of examples now, I think, in the following slides. Sorry, here you find some additional applications, biomarkers for pharmacodynamic evaluation, for example, CSF biomarkers in the evaluation of secretase modulators, effects on cholinergic neurotransmission, where QPS has also a rich track record, effects of antisense oligonucleotides on specific pharmacodynamic targets can be evaluated and are currently implemented in some of our clinical trials. Next slide. Pharmacodynamic studies can be performed based on it, and of course another outcome measure may be the prevention of conversion from MCI to AD in, next slide please, enriched patient populations, where you can actually define subgroups of subjects with MCI with specific markers, with, for example, specific ApoE4 genotypes that are very likely to convert to AD. So in other words, you increase the power of your study by recruiting that type of subjects in trials where the endpoint is diminution or decrease of conversion to AD. Next slide. Okay, also based on quite a lot of large data sets, among others from ADNI, we know that there is a succession of biomarker changes, and of course, clinical trials can also attempt to delay the biological progression over the different stages. Next slide. Okay, here another outcome measure, hippocampal atrophy, where we know actually based on historical data what the atrophy progression is, for example, in controls, and what the progression is in MCI. And so decrease of progressing atrophy can be considered as a potential outcome measure. Next slide. Okay, indeed, as we mentioned already, we had the aducanumab story with some positive news today. We had also the babineuzumab case, which we all followed during several years. This is a therapeutic strategy with passive immunization. Next slide. One of the outcome measures was actually the change in amyloid deposit using PIP-PET, and you see that indeed there are statistically significant decreases. Unfortunately, the compounds, the changes did not translate in clinical improvement, and the further development of babineuzumab was stopped as well. Next slide. Okay, I'm not going to deal with novel biomarkers, but I actually wanted to give a summary of the biomarkers that are currently used and studied extensively. There are many more, of course, now. There are a lot of biomarkers specifically focusing also on synaptic activity, synaptic density, and there is also a major development in peripheral biomarkers looking at, among others, plasma. Next slide. Okay, I would like to draw some conclusions so that Isaac Dendaas has sufficiently time to give his presentation. So, first of all, biomarkers contributed to diagnostic criteria for clinical practice and research, and the entire course of AD, from asymptomatic onset to the severe dementia form, is presently captured. That's important. Biomarkers are indeed increasingly important in AD research for the natural history and also for clinical trials as a tool. Application of criteria and biomarkers in clinical practice is also evolving rapidly. Next slide. We all witness that, of course. And then finally, I would like to state that biomarkers may contribute to enrichment strategies, as we discussed already earlier, that they can help us in monitoring disease progression, besides, of course, neuropsychological outcome measures, that they help us also in monitoring neuroprotection, which may be one of the outcome strategies or measurements, that the development of validated surrogate outcome measures have advanced the clinical research in this field, and actually the further development of those biomarkers has also led to the identification of novel drug targets. I think this is my last slide.
Next one.
And I thank you, of course. Voilà, that's it.
Okay, thank you, Peter, for the extensive overview of the latest development in CSF biomarkers. And I'm now going to start a short overview concerning the clinical capabilities. What do I need if I want to do a clinical trial and in early development? Next slide. The requirements for performing a clinical trial in AD, and of course, without question, good clinical practice, GCP, is an environment you absolutely need to perform clinical research. And I want to shortly discuss with you the regulatory environment in the Netherlands. That you need a science-driven organization, good collaboration with the local KOL, good environment for treating a vulnerable patient population. Patient recruitment, of course, is pivotal if you have a clinical research organization without their own patients. Early clinical development, where safety, tolerability, PK is routine, still an essential subject in early clinical development, and of course special techniques required, and of course we heard that already in the former talk. Imaging technology is pivotal. You need PET, MRI, and of course CSF is important, so you need a good experience in lumbar puncture or even serial CSF sampling. Cognition testing is needed. EEG is nice to have as well. Now I'm going to show you a couple of examples of these requirements we developed the last 10 years, also in collaboration with academic scientists like Peter Paul de Dijn. Next slide, please. Next slide. Yes, we are very lucky in the Netherlands for obtaining EC and regulatory approval in the Netherlands. We have a very fast system, and it is one of the fastest in Europe. There are parallel submissions to the health authority and the local ethics committee, resulting in clinical trial approval within 14 days. If you have a substantial amendment, you have a 48 turnaround time where you put your amendment towards the ethical committee, and the chairman will look at it, and if necessary, a specialist of the ethical committee. The CTA clinical trial application will be prepared and submitted by QPS, which is a possibility. We have a good relationship with the independent ethical committee. This ethical committee meets twice per month, and lately this year they started at 3 times a month, and submission is 11 days before the meeting. Now, after 1 or 3 times before the meeting, the PI of the study receives a memo of the pre-advisors of the ethical committee, and they can be answered by the PI during the EC meeting. The PI will join the meeting for answering questions, but will of course not attend the voting, and the CCMO will only perform a so-called marginal test. They check the ultravigilance and the completeness of CTA. Approval can then be given after 14 days as well. So a relatively fast process, which is already proven in approximately 300 studies with QPS, and of these 300 studies, only 2 were objected, and 2 of those were sent to newer meetings, and 1 of those was approved. So we have a good track record. Next slide. Now, as a CRO, a contract research organization, which is more or less specialized in clinical pharmacology, PK, can you go one back? With where you have no patients of your own, but good collaborations are needed. Now, for CSF sampling, we did collaborate with the Department of Anesthesiology already for more than 10 years. PET imaging with a very good PET imaging center 300 meters from our clinic here in Groningen. Recruitment of AD patients, you need Alzheimer centers, and Peter is, I think, a collaborator of the last 9 years already, but also outside the academic medical center. You can look for peripheral hospitals, and there are 3 hospitals we are working with, and we're also working together with another Alzheimer center, which is the VUMC Alzheimer Center and the Brain Research Center in Amsterdam. Next slide, please. You need a good environment for this vulnerable patient group, and hereby we started this year with a new clinic in the city of Leeuwarden, 50 kilometers here. It has a home feeling atmosphere, very important. The QPS site is small, it's only 1 floor with 8 rooms needs, and very flexible. We can have overnight stay in a hotel set at our center. The partner can stay as well. We have overnight care by trained QPS staff, research nurses and research physicians, and already our first study performed in May to August in 25 elderly subjects, including 2 lumbar punctures, MRI, PBMC isolation for white blood cells, and cognitive testing. Currently, we started with a Alzheimer patient study, including all these as well, but also doing EEG. Next slide. Now, you need a patient recruitment network. And of course, the collaboration with Professor De Dijn and Alzheimer Center in Groningen is a pivotal one. And you can see the blue spots where peripheral hospitals are in a network for collaboration. We also collaborate with the Brain Research Center and Amsterdam Neuroscience. We have a good peripheral general and teaching hospitals with memory clinics in the four northern provinces in the Netherlands, which are not that big populated as the rest of the Netherlands, but still good for more than 3 million people. Network takes care of referring. The network of Peter Paul De Dijn, which is well known with his network, will give us patients out of the periphery as well. Besides that, we have well-trained QPS physicians and nursing staff to perform these early phase Alzheimer's studies. Next slide, please. PET and MRI. As I already said, we have a long-standing experience with a good PET centers, more than 30 years of experience this PET center has. They have a strong recognition of industrial collaboration. They have their own PET and an MRI, 3 Tesla scanners located 300 meters from our clinical pharmacology unit. They also do animal PET, and QPS performs more than 10 studies, and I think we're getting close to 180 healthy volunteers, mainly healthy volunteers, but I think 20, 30 patients as well. The PET center has its own radiochemistry lab, so they can make their own radioligand. Their own cyclotrons, of course, takes care of the radioactive material, and they have PET SPECT and SPECT cameras, which are state-of-the-art Siemens collaboration products. Next one. CSF sampling nowadays pivotal in any trial. One back, please. Collaboration with the Department of Anesthesiology. They do lumbar punctures, but trained our research physicians as well. Lumbar punctures are performed in more than 10 studies. 2 or 3 lumbar puncture per subject is possible over a certain period of time, mostly 2 or 3 weeks. But if you need more, you can insert a CSF catheter for serial CSF sampling, performed by an experienced pain specialist of the UMCG. And we performed in healthy volunteers and Alzheimer patients up to 36 hours of sampling. These lumbar punctures and CSF catheters are associated with mild to moderate discomfort, but post-LP headaches were not reported yet in our studies. Now, asking the panels of subjects how they would rate your experience of pain or discomfort, and majority had none or mild problems like back pain or headache. And the question, would you do this again, this whole procedure? And that said something about the stamina of the Dutch citizens, perhaps, but 100% said, yes, we would do it again. Next slide. Cognition testing is something you need in most AD trials. We have experienced staff which can do all commercial testing batteries and use our MMSE composite cognitive battery and clinical dementia rating. Next slide, please. We have experience with early phase AD patient studies, 36 hours serial sampling, as I already said, an amyloid antibody multi-site study, antisense studies, not only in Alzheimer, but now also in Huntington disease, anti-inflammatory target studies also performed, and last but not least, a very exciting epigenetic target of which Peter also hinted, where the proof of concept cohorts are performed in collaboration with the Alzheimer Centrum in Amsterdam as well. And next slide. Thank you for your attention. Are there questions?
Yes, thank you very much for that insightful presentation. Now I'd like to invite our audience to continue sending in their questions or comments right now using the question window for the Q&A portion of this webinar. We've already received a bunch, so we'll start with what we have. Our first audience question is, have you examined neuroinflammatory markers such as IL-1B, TNFA, TGFB, MCP1, MIP1A, etc.?
I'm sorry.
So that was the question about examining of neuroinflammatory biomarkers in Alzheimer disease, I presume. Is that something we've done before, or?
Oh, whether we specifically focused on, of course, we did so, of course, in research context, but I personally did not do that related to specific therapeutic trials at this moment. So the answer is no, but of course we looked at several pro-inflammatory and inflammatory parameters, interleukins were studies, lipocalin and other substances, but not related to specific therapeutic trials as outcome measure. But Isaac maybe can correct me.
Yes, I mean we are currently talking with a client who wants to use these inflammatory markers in CSF to look if there is a inflammatory response, and if their compound is reacting on it. But we have not performed this study, but there is talk about this.
Yeah, maybe a very general comment. Sorry, I was focusing specifically on those clinical trials that we recently performed, but there are a lot of research lines focusing, of course, on tumor necrosis factor receptors in modulating substances, where there is also a lot of research being done on microglial activation as well in preclinical research, a lot of animal modeling as in the human condition, with a lot of interesting insights and progress recently being made.
Okay, I hope this answers the question. Next one.
All right, our next question is, don't the failures of anti-amyloid drugs suggest that maybe we don't really understand the neuropathology of AD?
That's a very good question.
Yeah, that's indeed actually a very good question, and everybody asks it, but I do not have an answer, but I'll give a very pragmatic answer. One, I think that indeed we know that amyloid plays a role. We know that tau plays a role. We know that inflammation plays a role, but we do not know which of those factors are the most important at which given moment. So, but that's the topic of a lot of research ongoing presently also in our center. Personally, I think that, we heard now just aducanumab, there are some promising additional analysis. So I do not know the details, but we are looking forward to find out what it is. But finally, I think there is consensus that we will have to target specific targets, diverse targets in one single individual, and that a lot of course, potential therapeutic effects you may achieve depends on the stage of the disorder, of course. And even as you know, the mild cognitive impairment stage is already a relatively advanced stage. But then, of course, you have the problem, can you start treating huge amounts of our population in a very early pre-symptomatic stage? So that's another question, practical and ethical question. But we know now that quite recently there are some additional insights that are somewhat more in favor of tau as a major target or focus. And we are focusing on that, but we still have to find out what it will yield.
Well, thank you very much for both those answers. Our next question is, what are the benefits of doing serial CSF sampling compared to single lumbar punctures?
There can be several reasons for it. First of all, you have to realize serial CSF sampling is far more intensive, of course, than single lumbar puncture. Nowadays, there might be a reason for doing serial CSF sampling over 2 or 3 lumbar punctures. One is if you want to know exactly what your pharmacokinetic in this central component is, in this central compartment is, might be very interested. You have a very strange PK profile peripheral, and you really want to know where is my Tmax in the CSF because that is as close as you get to the brain. That might be an interesting thing to study. Or you have a complicated biomarker profile which you want to look. Now, for instance, if you only have 2 shots in a profile, it's easy to miss, for instance, a Cmax or a Tmax. But if you do a serial where you sample, let's say over 24 or 30 hours, let's say every hour and later on every 2 or 3 hours, that is something which might give you extra information about your compound, which might be vital for your pharmacodynamic.
Yeah, sorry, I fully agree. Yeah, I mean, it's quite used also as a pharmacodynamic outcome measure, of course. And if you focus on tau, for example, yeah, you want to lower your tau. And for that, of course, repetitive sampling is, of course, indicated.
Thank you. And our next audience question is, can you say a few words about early onset AD and how or if it differs from AD in the elderly?
Wow. This is a very difficult question. And this is actually a question I ask to my students quite often when they study the pathophysiology of AD. As a matter of fact, the neuropathological findings are very comparable. The timelines may be somewhat different in terms of microglia activation and amyloid deposit, but overall, at a given stage, when you look at the brain, they are similar, whether it's early onset AD or late onset AD. The other aspect is, of course, that the amyloid accumulation has different reasons, while in the early onset AD, it's most of the time an increased production of amyloid beta 142. So there are some differences, and that's why, on one hand, we know those mutations very well, like, for example, the APP, presenilin 1 and presenilin 2 mutations, and we know also the progression of the biomarkers, specifically in those families, in those autosomal dominant entities, but they may not always be, how to say, one should not always be able to extrapolate to the more sporadic AD in the elderly. So maybe sometimes we make a mistake there. And most probably the pathophysiological mechanisms may still be different in early onset versus late onset Alzheimer's disease.
Thank you very much, Professor Peter Paul, for that answer. Our next audience question is, I want to perform a clinical study with an anti-tau compound. Which biomarkers would you recommend?
Well, Peter, I would say tau and p-tau would be a good candidate.
Right. Yeah,
and then in the CSF, I presume?
Yeah, and you could do that indeed in CSF. I mean, in plasma or peripheral biofluids, it doesn't work, certainly not at the moment, but indeed CSF, and one could consider also tau PET imaging, of course, which we presently also apply in some of our ongoing studies. So, but monitoring CSF, tau and phospho-tau levels would be, of course, the most practical approach.
All right, thank you. That brings us to our next audience question. Are there peripheral biomarkers I can use if I am going to perform a study with a compound which act in the CNS inflammatory cascade?
I think this is a very good question. And I think what I see with customers as well, that it is, of course, sort of holy grail one wants to search. There are indications that with the help from white blood cells isolated with PBMC isolation, there is exploratory work on that, but I think the judges are still out on that, and definitely nothing up to now has come to a conclusive result. although I must say there's a lot of activity in that area. What do you think, Peter?
Yeah, I do think so, and I think that presently focusing on those specific cell lines, a lot of innovative research is taking place also in trying to classify people, among others with AD. So indeed, that's promising.
Thank you both for those answers. Our next audience question is, we want to perform a study in a population of AD patients which are in a moderate to severe state of disease. Is such a population recruitable, and how is the situation with the patient's ability of consent?
Peter.
Well, yeah, so that was my first thought or reaction was, and I think Isaac agrees that indeed the moderate or more advanced stage is, let's say, relatively easy to recruit because, you know, the very early stages with very mild cognitive impairment, yeah, do not last that long. The mild AD or moderate AD much longer, but then you run into the issue of competence of the subjects, the preparedness or competence to still give consent and decide. And that's an issue, yeah, which is not easy. You know, of course, that there are some questionnaires, if I'm not mistaken, MacArthur and other ones, which are a kind of questionnaires that allow you to evaluate the competence of a subject to decide on accepting a treatment or participating to research. So there are criteria, but some people are somewhat resistant to enroll subjects in a more advanced stage. Although I would say that's actually a form of discrimination. If somebody says, look, if I'm more advanced, I'm still willing that you attempt new therapeutic strategies on me as a person or on my body, that right should not be denied. But I mean, it's a little bit an ethical sensitive issue.
It is, and as a representative of a contract research organization, I find that very difficult if the person himself has difficulties giving a consent, because this is different than in an academic setting, I would say. There are possibilities definitely here, and that's the same with, I would compare that with pediatrics. Pediatrics is a very difficult area as well, because the young child cannot give his consent. His parents have to give his consent. There's a whole discussion about that. And in pediatrics, I have not seen that much of a discussion in dementia this way. And perhaps it's a good time to start up a discussion like that.
Well, for sure, in the past, we have studied a lot of substances in the advanced stages. I mean, to give an example, memantine, for example, which was introduced on the market, was actually indicated for advanced stages, looking at people with an MMSE score of 12 or below. And indeed, recently, we are not conducting those studies anymore in that specific population.
Well, thank you very much for those answers. It looks like that's all the time we have today to answer questions. If we weren't able to attend your questions, please use this contact information showing on the screen. QPS may follow up with you after the webinar. Thank you everyone for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now please join us in thanking our speakers Isaac Dindas and Professor Peter Paul Dendane. You may also register for the next QPS webinar using the URL now showing in your chat box. We hope you found this conference informative, and have a great day, everyone.
Thank you.